Hello and welcome to today's talk on color vision in our series on physiological psychology. We've already talked in previous sessions about the physiology of vision and neural processing in the visual pathway. Specifically today I want to talk about color vision. We'll talk today first about Trichromatic Theory and its sort of close relative opponent-process theory, and talk about how these two theories sort of combine together to help us describe the way in which our three cone types and rods combine to allow us to process color vision. We'll talk then about some color vision deficiencies, talk a little bit about cortical color vision, and then finish up with a discussion of additive and subtractive color mixing. The trichromatic theory was proposed by both Young and Helmholtz. It's often called the Young-Helmholtz theory of color vision. According to this theory, color vision depends on three different receptor mechanisms, each with different spectral sensitivities. They discovered that a normal color observer requires a light of three different wavelengths to match colors specifically. In this particular case, they used a test field and a comparison field, and they could mix three different wavelengths of light to get it to match this 500 nanometer light on the left. What Young and Helmholtz proposed in their trichromatic theory is that the three cone photopigments are responsible for these three different wavelength phenomena in creating color vision. Remember, there are different absorption spectra for the three different cone types. The short wavelength cones absorb light best at about 420 nanometers. With medium wavelength cones, they're at about 520. And the long wavelength cones there at about 580. So this different absorption spectra of the three different cone types is responsible for our ability to process color vision. There's also some really interesting research on the genetics of each of the different opsins and the amino acid sequences they express. So if you look here at the left, it looks at the short wavelength cones versus rhodopsin. Very, very different expressions of proteins versus the medium wavelength versus short wavelength, also very different. But the long and medium wavelength opsins are really quite similar. And what can often happen is those two protein sequences are so similar that you can end up with what's called anomalous trichromacy, in which the absorption spectra are so close together that it's difficult to discern red from green. So if we start here and look at the medium and short wavelength cones, you can see there's a huge amount of differences. These filled circles indicate places where there are differences in the amino acid sequence for the opsins. But if you look at the medium versus long wavelength cone types, they are quite similar. And as I said, a little bit of alteration in these amino acid sequences, and you can end up with absorption spectra that overlap a great deal more than they are supposed to. So one of the things we know about the physiology of trichromatic theory is that two pigments are sufficient for color vision, but one is not. And we call this the principle of univariance. One pigment can respond only to the presence or absence of light within its absorption spectra. It can't tell us how far away the so we need two photopigments for processing of wavelength. This allows us to sort of allows our eyes to be able to calculate and understand where wavelengths of light are coming from by the varying responses to the different cone types. So two pigments are sufficient for processing color information, but three pigments allow for superior color processing. And in fact, we know some people, particularly some women, may have potentially four different cone pigments. And we know some species of animals, particularly raptors, have more than three cone types, a lot of avian species. And there is recent evidence that the mantis shrimp has up to 12 different cone types, so they have really superior color vision. Well, we certainly know that the three cone types are required for normal color vision, but that's not sufficient to explain everything we know about color. So opponent-process theory was originally proposed by Herring. This essentially came along because trichromatic theory is insufficient to describe some of the phenomenon involved with how we process color, and one of these has to do with color after images. So I'm going to present a series of images followed by a blank white slide. And so what you'll do is you'll stare at the color image until the slide goes on to the next blank screen. And then what I want you to notice is how what happens to the colors, what changes from red, what changes, what was red, what does it become, what was green, what does it become, etc. So we'll start with this quick instruction screen. So stare at the image in the next screen until the next blank screen appears, and then continue staring at the screen to see the image. And of course, if you have trouble with this, you can pause and back up and move forward on the video. So we'll start here. Be sure you try to stare right at that fixation point, that black dot in the center, and try not to move your eyes. Perfectly fine to blink, but try to keep your eyes focused on that one central point. You should be getting an after image, and you should notice the locations of those colors have shifted. That is, what was once red is now green, what was green is now red, what was blue is now yellow, and what was yellow is now blue, indicating that there is an opponent process involved. We'll take a second to let that clear, and then we'll show you yet another one of these. Again, stare at that center fixation point. Okay. Again, the color fields have reversed. What's happening in these instances is we've adapted out the color on the screen. Now remember, your neural systems are constantly firing. So the sort of blue system and the sort of the system responsible for blue and yellow and red and green, they're sort of constantly firing anyway, but they're always in balance with one another. What happens when you adapt out one of those colors is once you take that color away, its color drops below its normal responding, and what happens is then, because of that difference, because the red, what was responding to red, has now dropped below its normal response rate while those neurons recover, that's during that refractory period, the opponent process takes over, and that's what you observe. So we'll do one more. I believe there's one or two more of these. So again, just stare at that center of the screen. This takes about 20 or 30 seconds. And again, you should get a nice Union Jack British flag. Wait for that to dissipate a little bit. So what this demonstrates is that while certainly those different cone types are necessary for color vision, they do work in opposition to one another. And I'm going to skip through these next couple and move on to talk about how this all works. So some colors are what we call mutually exclusive. So red and green are an example of that. There are no sort of reddish greens or bluish yellows. And we call this the mutual exclusivity of colors, which indicates that there are independent neural processes involved in those colors, that red and green work in opposition and blue and yellow work in opposition. So we believe that there are actually three opponent channels: a blue-yellow channel, a red-green channel, and a luminance channel, which is simply black versus white. And this is wired up by the M and L cones creating a red-green circuit, and then the S cones with the M and L cones creating a blue and yellow circuit. So what this indicates is those S cones are necessary for any blue-yellow perception, but only the M and L cones are required for red and green processing, whereas both the S, M, and L cones are all necessary for processing blue and yellow. So this is kind of how this all wires up. We have an achromatic or brightness system that is simply the sum of all three cone types. Then we have a opponent process for the long wavelength and medium wavelength cones, and then we have another opponent process where the long wavelength and medium wavelength cone types add together and then are in opposition to the short wavelength cone types. So normal color vision then is described by the relative contributions of each cone type, and in fact this relationship can be mathematically described. And there's a great deal of work going into this where behavioral neuroscientists actually look at the density of different cone types at different parts of the retina and see how color vision changes across that distribution. So as we start having fewer S cones, fewer M cones, fewer L cones, as we move away from the fovea, you can actually mathematically describe how those different cone types contribute to normal color vision. We won't describe that here, but there are certainly plenty of places you can go to look for that kind of information. To finish up our discussion of color, we're going to talk about color deficiencies, followed by talking a little bit about cortical vision, and then finishing up with color mixing. I want to talk now about color vision deficiency. This is improperly often called color blindness. And in fact, the only really genuinely colorblind people are those who have achromatopsia, which is damage to the cortical centers of the brain responsible for processing color. Most people are what we call a dichromat. That is, they're missing one of the color pigments. The first of these is called protanopia. These are people who are missing the long wavelength photopigment. Deuteranopia is an individual missing an M pigment, and then tritanopia are people missing the S cone photopigment. The first of these two, protanopia and deuteranopia, are actually sex-linked traits. That is, they are the gene that codes for the opsins in the medium and long wavelength photopigments is in the unmatched part of the X chromosome. So males are far more likely to have this kind of color deficiency than are women. Women, in order for them to have a protan or deutan defect, have to be missing the gene coding for the long or medium wavelength photopigments from both parents. So they have to inherit it from both parents, whereas males only have to inherit it from their mothers. Tritanopia is the gene coding for the short wavelength photopigment is on a different part of the genome. And there's a whole sort of interesting theory behind why that happens, because we think we developed this ability to see blue later in our evolution. So it's in a whole different part of the genome. And so that is not sex-linked, and it's also relatively rare. The other kind of color vision deficiency you might run into is someone who's called an anomalous trichromat, and I actually happen to be an anomalous trichromat. So I'm about to show you the Ishihara Pseudoisochromatic plates. I can't tell you what it's supposed to look like because I can't see them, but you should be able to see certain shapes, numbers, etc. In the following Ishihara plates. And if you're concerned about this, there are plenty of online color vision tests, and you can certainly speak with your ophthalmologist or optometrist to get some in-person color vision testing. Color vision testing is difficult to do on computers because everyone's settings are a little bit different, and so we're not always certain that you're getting the right kind of color vision testing. So an anomalous trichromat, anyway, as I was saying, are people who have three photopigment types, but they have different absorption spectra that aren't independent enough, that aren't spaced far apart enough to be able to determine red from green. And so it's very difficult for me to see red from green. So we're going to take a look at the Ishihara Pseudoisochromatic plates. There's also the Farnsworth-Munsell 100 color arrangement test. There's the Neat's color vision test. These are all different ways in which you can have your color vision tested. So we're going to take a look at the Ishihara Pseudoisochromatic plates. So this is a baseline plate where you should be able to see the number 25. I'm going to present these relatively quickly for you to just look at and to get a sense of what they look like. So here we have, as I said, I can't tell you what you're supposed to see in all of these because I can't see things in all of them. But you get the sense of how this is supposed to look. This is one where I can see a 3, but there's a second number, I believe, that I can't see. I can see nothing in this one. Same with that one. There's an 8 there. Yeah, again, nothing I can see. So damage to the cortex is going to result in achromatopsia. And this, what happens here is people actually genuinely have black and white vision. Other damage to the cortex can cause what's called color agnosia, which is an inability to recognize color. The cortex is lined up in an interesting way. When it comes to color vision, there are actually receptive fields that are antagonistic to different colors. So one of the reasons why someone like myself, I can see red and I can see green, but when they're lined up right next to each other, as in those plates, I can't discern red from green very well because of the receptive field nature of our color vision. So that's a little bit about cortex. There's certainly lots of important information. Again, this is usually in what we call the ventral stream or the wet pathway, where most of our color vision processing occurs. To provide a little practical perspective, I want to talk a little bit about additive versus subtractive color mixing. There are two different ways to sort of mix color. Most of us are familiar with subtractive color mixing. In fact, that's most of what color mixing we've done all our lives is actually subtractive color mixing. So pigments such as paints obey subtractive color mixture rules because what you're doing is you're subtracting out more wavelengths of light. So when you have blue paint and yellow paint and you mix them together, the reason it looks green is because the light that's now being reflected back to you is green because you have subtracted out some of the other wavelengths. Whereas if you add a blue light to a yellow light, you get a white light. And in fact, that's how many light fixtures work, is they actually don't have a full wavelength, the full spectrum of light. While it looks white, it's actually mostly blue and yellow light. So additive color mixing occurs when you add lights together. This is how your computer and your TV work. People who are involved in theater know a lot about this because we're often talking about mixing different colored lights and putting them on specific kinds of surfaces and trying to get a specific look based on both additive and subtractive color mixing. So if you look at, if you're trying to make a color, for example, and that's where this comes from, in Microsoft Word, you can just simply play around with the contributions of red, green, and blue. Everything on your color is on your computer is red, green, and blue. So you can see here I've maxed out the red and the green and zeroed out the blue, and we end up with a yellow light. So red plus green becomes yellow. Remember, this is the yellow-blue channel. With no opponent process, no minusing out blue, we end up with yellow. So blue and yellow equals white. Red and green equals yellow. So what happens if we have red, green, and blue? Well, we end up again with white light. Looks something like this. Subtractive color mixing occurs when we mix paints. So paints are subtractive color mixing because we're subtracting more light when we add one paint to another. So this is what that looks like. So we might have blue paint. That's reflecting out mostly blue light. We have yellow paint that's reflecting back mostly yellow light, maybe with a little bit of green. So we mix those two together and we end up, the only thing that's being reflected back is green paint. Now the most important thing to understand about subtractive color mixing, and again this is where I think things become important, because as you start owning homes and start trying to come up with paint colors for your houses, it's important to understand that you're doing both additive and subtractive color mixing when we're talking about painting, because the paint that you're looking at Home Depot or Lowe's is underneath the lights that are at lows. So, but when you take that paint outside, or when you take that paint into your own home, you have different light sources that may have a different set of wavelengths associated with them. So those colors may look very, very different under a different light source. So you always want to make sure when you're picking out paint that you take it home and try it out first and try to see if it looks like what you want it to look like in your specific lighting circumstances. This is one of the reasons why when you buy something at a store like Hollister that has very dim illumination, it may look very, very different when you take it outside into the bright light. Now, the principle of color constancy states that it will mostly look the same as you move, say, from bright light inside to bright light outside, because we have sort of this cortical color mechanism for how we process color. But there's only so much that color constancy can do. So we're talking about a complete and total change in illumination. We oftentimes see a complete and total change in our perception of color. And again, that has to do with both additive and subtractive color mixing. So that's an introduction to color vision. There's a lot more detail we could get into in this, but I wanted to provide a basic introduction to the topic. And next we're going to be switching gears and talking about the auditory system. So stay tuned for that.